Okay, welcome everybody. This is our first podcast of the semester, and we're going to be dealing today with special senses. And so we'll start with a discussion of general senses, then we'll move into the special senses, which are defined by the presence of a special sense organ. So the special senses include olfaction, gustation, audition, and vestibular sensation. Olfaction, meaning the sense of smell, gustation, taste, hearing is audition, and vestibular sensation being your sense of balance. Special sensory neurons detect light, chemicals, and sounds present in the environment and convert or transduce them. These stimuli are changed into electrical signals that can be interpreted by the central nervous system, and this is very important to understand. Our body has to become aware of stimuli which happen inside and outside the body, and that awareness is coordinated primarily through the nervous system. The nervous system, however, only understands electrical information, and so it's the job of general and special sense organs to turn physical changes into electrical information that can be interpreted by the brain and the brainstem and relayed up and down the spinal cord. Many special senses rely on receptors that are not neurons. General sensory receptors are the receptory ends of sensory neurons, while special sensory organs that are confined to the head convey information that travels along axons of various cranial nerves. The general sensory information travels on the axons of both cranial and spinal nerves. And so you can see here how the general senses and the special senses operate. These are all indications of physical change. They are going to produce an electrical event on the afferent neurons that are going to bring this information back into the CNS. And that event, more often than not, is a depolarizing event in which ion gates are opened that allow positively charged ions, which in most cases are sodium, to flood in to the nerve cell or into the receptor cell, generate a depolarizing current, and if that change is sufficiently strong, that can produce an action potential. And so you can see how it's set up with general senses. It's usually a direct event that happens on the neural endings, and these endings are going to be modified with regard to their modality. In other words, touch is going to be governed by mechanoreceptors that generate a depolarizing action, pain nociceptors, which are, generally speaking, free nerve endings that can respond to extremes of chemical or thermal or physical stimulation, and temperature are a type of receptor that is going to respond to a change in heat content, opening or closing ion gates accordingly. The special senses are going to impinge on cells that are designed to respond to their modality. For taste, the binding of tastants is going to trigger a change in membrane potential. These are found in the taste buds of the tongue. For light, photoreceptors are going to respond to color or light intensity, generating now a change on the membrane potential of the photoreceptors that is going to be relayed ultimately to afferent neurons that are going to travel via the optic nerve back to the thalamus of the brain and from there to the occipital lobe. And sound is the purview of the cochlea, as we're going to find out. The cochlea has specialized receptor cells that respond to pressure waves known as sound. That's what a sound wave is. It's a pressure wave conveyed through gas, and it causes vibration to occur, and that opens ion gates that eventually going to result in the production of an action potential that's going to be sent via the 8th cranial nerve back ultimately to the temporal lobe of the brain. And head movement is the responsibility of the semicircular canals in the vestibular apparatus, whose receptor cells, instead of responding to sound waves, respond to physical changes such as gravity an angular movement. And then smell, you can see, is again a direct impingement on a bipolar neuron that is going to produce an electrical change on the olfactory bulb, and that's going to be relayed via the olfactory tracts back to the olfactory cortex, which is thought to lie near the temporal lobe. And this is the only special sense that bypasses the thalamus on the way back to the brain. A way for you to determine whether we're dealing with a type of transduction that has to do with direct changes on neurons versus a change in a receptor cell that then talks to a neuron is to look at the anatomy of the receptor apparatus. Look for receptor cells that are first to increase or decrease the amount of neurotransmitter they release onto a neuron. This might come first in a pathway, as in olfaction, or somewhere in the middle, as in hearing. Once you find these cells, look for the stimulus that causes them to alter the release of neurotransmitter. Is it a chemical, sound, or light? Regardless of the type, the stimulus will alter the membrane potential by opening or closing an ion channel, and this changes the release of neurotransmitter by receptor cells. The details of the transduction might look a little bit different for each special sense, but the fundamental process is very similar. Okay, so we have basically an apparatus that's designed for its location and modality to produce a electrical signal that can be the result of the opening of ion gates that ultimately will produce an action potential that will be relayed back to the central nervous system via a spinal or cranial nerve. So we'll start with olfaction. The olfactory system allows for detection of odorants in the air. It transduces them into signals that are perceived as odors. Olfaction is initiated at the olfactory epithelium, which is a small region of specialized cells in the nasal cavity. There's three types. Olfactory neurons, or olfactory receptor cells, are modified bipolar neurons. They have chemoreceptors that detect chemical substances perceived as odors. Basal cells are stem cells that continuously replace olfactory neurons and have a limited lifespan of between 1 and 2 months. Supporting cells are columnar cells that surround and protect the olfactory neurons so that they can function. The olfactory epithelium and its anatomical structure includes the olfactory nerve, that's the first cranial nerve, which are the combined axons of the olfactory neurons. The olfactory bulb, which is a structure that sits atop the cribriform plate of the ethmoid bone and is inferior to the frontal lobe of the brain. And the olfactory tract, that's composed of axons that exit the olfactory bulb and travel to other regions of the central nervous system for interpretation. And so you can see where this lies. This is unique in that this represents one of the few pieces of central nervous system tissue that is exposed to the external environment. The olfactory bulb and the olfactory epithelia are out there in the open air, and as a result, the regenerative property of olfactory bulb neurons and olfactory epithelial cells is unique in the central nervous system. They have to be regenerated because they wear out. This is one of the few cases in the central nervous system where we see neurons that have the ability to regenerate. They're actually produced in the occipital lobe of the brain, and then they travel via a pathway called the rostral migratory stream until they reach the olfactory bulb. And when they get to the olfactory bulb, they stop and they differentiate, and they provide our sense of smell for a couple of months. Now, how is it that we change odorants into information that can be understood by the central nervous system? Well, odorants in inhaled air are detected by olfactory neurons. These are chemical stimuli that are transduced to electrical signals, which are transmitted to different regions of the brain for identification. The activation of olfactory receptors involves odorants that are dissolved in the mucus that surrounds the olfactory neuron cilia. Odorant binding proteins transport the odorants through the mucus to receptors on the cilia of the olfactory neuron. Next, once the binding of the odorant to its receptor takes place, we activate G proteins. The activated G protein triggers the enzyme adenylate cyclase to convert ATP into cyclic AMP. The cyclic AMP opens the ion channels that allow the sodium and calcium ions to enter the cell. This produces depolarization and action potential generation if we reach threshold. And once an action potential is produced, the odorant has been transduced from a chemical to an electrical stimulus. This signal then travels via the olfactory bulb to the olfactory tract and ultimately back to the olfactory cortex. Now remember, this is an example of a second messenger system. Okay, the utility of second messengers is that they serve to amplify a tiny chemical or physical change into a large intracellular signal. Okay, and this happens because there's not a one-to-one correspondence at each step in the cascade. When we activate, for instance, a receptor on the surface of an olfactory bipolar neuron, that activated receptor can activate several G proteins, which can activate several adenylate cyclases, which can produce lots of cyclic AMP, and that in turn can open lots of calcium and sodium ion gates, allowing them to flow down their concentration gradients from the outside to the inside of the cell, and that's what produces the depolarizing stimulus. And so you can see that at each step we magnify the signal. The other important part that's critical, the utility of the cellular signaling system, is that it's easily turned off because all we have to do is disengage the first or second steps in the cascade. The result then is that the cascade will shut down as enzymes that degrade the intermediates, which are always present inside the cell, manage to, for instance, break down the cyclic AMP and any other intermediate molecule that might be involved in the cell signaling, and thus the signal shuts off very quickly. Okay, we've seen cascades in several places. We see intracellular cascades as a method of cell signaling. We see extracellular cascades in phenomena such as blood clotting. The olfactory pathway consists of neural signals that carry olfactory information and travel to various regions of the brain for detection and odor identification, as shown in the figure. The axons of the olfactory neurons carry olfactory stimuli to the olfactory bulb in the central nervous system. The olfactory neuron axon terminals synapse with dendrites of mitral cells, which are neurons that lie in the olfactory bulb. The mitral cells of the axon form the bulk of the olfactory tract. This travels between the olfactory bulb, with primary olfactory cortex being the target in the temporal lobe. The olfactory tract is the only sensory pathway that bypasses the thalamus, and it's also considered to be part of the limbic system, the emotional component of our brain. The primary olfactory cortex is responsible for the awareness and identification of an odor. The amygdala, the hippocampus, the hypothalamus, as well as components of the limbic system, receive information from the primary olfactory cortex, and this evokes emotional and visceral responses to odors. It also can evoke certain memories. It's thought that the sense of smell is one of the strongest triggers of memory known. Now, a little word on anosmia. Sometimes people lose their sense of smell, and that's what the term for this disease indicates. Anosmia can be serious. People with anosmia may not be able to detect smoke or odor of spoiled food. They may suffer from malnutrition. Remember that olfaction contributes to taste, and so food loses its appeal. A lot of conditions can result in anosmia or hyposmia. Anything that blocks air from reaching the olfactory epithelium will interfere with olfaction, such as nasal polyps, a deviated or fractured nasal septum, a swollen respiratory epithelium from respiratory infections or allergic reactions, and even the common cold. In these cases, the anosmia is temporary and olfaction returns once we restore flow. but it can be permanent. Head injuries that damage neural pathways at any point can disrupt olfaction. The olfactory nerve is particularly vulnerable to fracture of the cribriform plate. Neural degeneration caused by Alzheimer's and Parkinson's can impair the sense of smell, and decreased olfaction may be an early sign of some of these neurodegenerative disorders. Hyposmia also develops naturally with aging. Fewer olfactory neurons are replaced by basal cells in the olfactory epithelium, and this process occurs gradually but becomes especially noticeable after age 60. And this is one of the reasons why in the elderly we often have to remind them to eat. Food becomes less appealing as we lose our sense of smell and taste. The result is that if we don't enjoy eating, we don't eat, and if we don't eat, we lose our fuel source, we lose our energy source, and that accelerates degenerative processes throughout the body. Okay, next special sense we want to touch on is taste. The gustatory sense involves chemoreceptors stimulated by chemicals. It's a complicated process involving olfactory chemoreceptors, thermoreceptors, and nociceptors, in addition to gustatory chemoreceptors. Now, for those of you who don't remember, okay, olfactory chemoreceptors are going to provide our sense of smell. Thermoreceptors respond to temperature, nociceptors to pain, and gustatory chemoreceptors to taste. It begins with the stimulation of specialized receptor cells called taste buds, which are clusters of receptor cells and supporting cells scattered around the tongue and other surfaces of the oral cavity, such as the throat and portions of the hard and soft palate. Each taste bud is associated with a sensory neuron that carries information to the central nervous system. If we look at the dorsal surface of the tongue, as shown here, we see that it's covered with projections called papillae. These can be further classed into 4 groups based on their shape. Valate, which are the largest of the 4 classes, they have a domed shape and contain hundreds of taste buds, and you can see them at the back of the tongue here. Okay. Fungiform papillae, which are mushroom-shaped and have only a few taste buds. Foliate papillae, which are ridges on the side of your tongue that have taste buds in childhood and are leaf-shaped, hence the name foliate. And filiform papillae, which are long, thin cylinders scattered across the tongue that lack taste buds and have sensory nerve endings that detect texture and temperature of food. Taste buds are usually located on the lateral surfaces of the papillae and have 3 cell types. The gustatory cells are specialized epithelial cells that contain microvilli that project into a small opening on the papilla called a taste pore, and they display receptors that detect different tastes. And they're associated with sensory neurons that carry taste to the central nervous system. These nerves, these cranial nerves that are associated with these neurons, include cranial nerves 7, 9, and 10. Okay? So that's facial, okay, then glossopharyngeal, and then vagus. Basal cells are stem cells that continuously differentiate into new gustatory cells and have a lifespan of about 2 weeks. Supporting cells surround and physically support the gustatory cells but have no role in taste sensation. And so you can see in the diagram the lateral margins of the papillae where the taste buds are contained, and you can see the gustatory cells, you can see the supporting cells, and of course the basal cells. And there's the taste pore with the microvilli. And there's what an actual histosection looks like. The sense of taste involves signal transduction of chemicals in food into electrical signals that can be sent to the central nervous system. Taste sensations rely on the detection of 5 classes of chemicals. Sweet tastes are elicited by simple sugars such as glucose and fructose. Lead paint and ethylene glycol also have a sweet taste. This can cause accidental poisoning. The taste is related to the shape of the molecule. It binds the same receptor. Sour tastes are produced by hydrogen ions, such as in citric acid in lemon juice and other citrus fruits. Salty tastes are elicited by the presence of metal ions such as sodium or potassium, and bitter flavors are produced by nitrogen-containing compounds found in rancid or poisonous substances. Examples include things like alkaloids that are found in a lot of plant materials. We have an adverse reaction to bitter flavor. We've been selected for this. As a result of natural selection, we avoid bitter-tasting compounds that are likely to be noxious or poisonous. Umami is the taste associated with meat or broth, and it's produced by glutamate and other amino acids. The detection of taste begins when a chemical stimulus binds to a receptor on a microvilli of a gustatory cell. Taste receptors are classed by the substance they detect, with only one type of receptor associated with an individual gustatory cell. The activation of taste receptors begins with the substance dissolving in the saliva before it can reach the taste bud, where it can be detected as a gustatory stimulus. The following steps occur once a taste stimulus reaches a gustatory cell and is transduced into electrical signals. We see a change in ion movements. This produces depolarization of the gustatory cell's plasma membrane, an example of the gradients core principle that we talked about in ANP1. Next, the depolarization of membrane opens the voltage-gated calcium ion channels, causing calcium ions to enter the cell. The calcium ions trigger the release of neurotransmitter, and this produces an action potential in the axon of a sensory neuron in cranial nerve 7, 9, or 10. The gustatory pathway follows the afferent neurons that exist in 7, 9, and 10. These are the facial, glossopharyngeal, and vagus nerves. They carry taste stimuli from the tongue to the central nervous system, and they terminate in the solitary nucleus in the medulla oblongata by synapsing on central sensory neurons. Axons from the solitary nucleus synapse onto neurons in the thalamus. They send their impulses to the primary gustatory cortex in the parietal and the insular lobes. Okay, so you can see here the primary gustatory cortex. There's the insula down here. It's that hidden fifth lobe that is sandwiched underneath and between the temporal and parietal and frontal lobes. And then you can also see it involves a portion of the parietal lobe as well. Next, the taste signals travel to either the inferior region of the frontal lobe for integration with visual and olfactory information, or to the limbic system, and this provides our emotional reaction to taste. Okay, next we want to address the organ of vision, the eye. The eyeballs are located in the orbits on either side of the nasal cavity in the skull. Each eyeball occupies the anterior third of the orbit. Adipose tissue provides protective cushioning around the eye. It keeps the eyeball in proper position and occupies the posterior two-thirds of the orbit. Note that the orbit has several accessory structures. They have various roles in protection and function in the eyeball. The eyelids are two thin folds that cover the anterior region of the orbit and prevent access by foreign objects and distribute tears during blinking. The eyelids have the following anatomical features: tarsal plates, which are thin pieces of dense, regular, collagenous connective tissue that reinforce each eyelid. Tarsal glands that are modified sebaceous glands located with tarsal plates that secrete oils, preventing the eyelids from sticking together. Medial and lateral commissures, known as the canthi, where the upper and lower eyelids meet. The lacrimal caruncle, which is a fleshy structure found at the medial commissure. It has sebaceous glands that secrete a whitish lubricating substance. The orbicularis oculi muscle closes the eyelids, while the levator palpebri superioris elevates the upper eyelid. And you can see also the muscles that move the eyeball: superior oblique and superior rectus, the lateral and inferior rectus and inferior oblique, and not visible in this figure, the medial rectus. The eyebrows and eyelashes are protective structures with the following features. Hairs found on the ridge of the brow form the eyebrows and prevent perspiration from getting into your eyes. So they're like your nature's sweat bands. They reduce glare from bright light and are important for facial expression. The eyelashes are stiff hairs that are found on the edges of eyelids associated with nerve endings that blink when objects touch them, and this decreases the aspect of eye injury. Conjunctiva are thin, continuous epithelial membranes. They line both the posterior surface of the eyelid and the anterior surface of the eyeball. This is a translucent membrane which have tiny blood vessels that can be seen. And the palpebral conjunctiva cover the eyelid's inner surface. They turn back on themselves to form the bulbar or the ocular conjunctiva, and this covers the white part of the eyeball. The lacrimal apparatus is designed to keep the surface of the eyeball moist so that we can flush pathogens and particulates out of the eye. More importantly, keeping the cornea, which is the transparent skin on the front of the eye here, moist allows it to get oxygen from the air so that it remains a viable tissue. Okay? Note that the tears are produced by the lacrimal glands. They flow across the surface of the eyeball, draining into the lacrimal caniculi, which then drain into the nasolacrimal duct, which drains into the nasal cavity. And this is why when you cry, your nose runs. The lacrimal apparatus produces tears that drain from the eye. The lacrimal gland is in the superior lateral region of the orbit behind the conjunctiva, releases tears and mucus into ducts that enter the conjunctival sac when stimulated by autonomic neurons. This lubricates and washes away debris. Blinking sweeps the tears medially and inferiorly across the eye, where they can be drained into passages that lead to the nasal cavity. The tears first enter the tiny openings on the medial edge of each eyelid called the lacrimal puncta, which lead into the lacrimal canaliculi, which empty into the lacrimal sac. This sac is located in a small depression in the lacrimal bone that drains into the nasolacrimal duct, which travels through the lacrimal and maxillary bones and opens into the nasal cavity just below the inferior nasal concha. Now, it's critical that this apparatus work. You may have heard of a condition known as dry eye. What happens in dry eye is that we reduce tear production. The result then is when you blink, you can tear the cornea off the front of your eye, and this is extremely painful. The fix are medications such as artificial tears, as well as medications designed to increase the activity of the lacrimal glands. The extrinsic eye muscles are designed to move the eyeball around in its socket. These 6 muscles originate from the orbit, and they insert onto the outer layer of the eyeball and allow for precise movements. The superior, inferior, and lateral and medial rectus muscles extend from a common tendinous ring on the posterior wall to their respective insertion sites on the eyeball. Each rectus muscle moves the eyeball primarily in a direction for which the muscle is named. Superior and inferior rectus muscles move the eyeball medially in addition to superiorly and inferiorly. The superior oblique travels from the posterior orbit along the medial wall through a loop known as the trochlea before inserting into the superolateral aspect of the eyeball. Contraction depresses the eye and moves it laterally. Okay, so this allows you to look down and to the side for each eye. Okay, now again, words mean things. The trochlea is the name for that little pulley that the muscle moves through in order to attach to the eyeball and move it, and it's innervated by the trochlear nerve. And you want to make those kind of connections so that you can understand not only the innervation of that muscle, but how we might test for a problem with that particular muscle. Okay, so if you indicated to a patient to look down and to the side, and they couldn't do it, that would indicate a problem either with the muscle itself or the innervation of that muscle, and that would tell you that the trochlear nerve is experiencing some kind of problem. So remember the trochlear nerve in old Olympus towering tops, that's cranial nerve 4. Okay, is one of several nerves that move the eyeball, right? The trochlear, the oculomotor, and the abducens are the big three. The inferior oblique muscle originates from the medial floor of the orbit to insert on the inferior lateral aspect of each eyeball. Contraction elevates and moves the eye laterally. Precise movements of the eye require a great number of small motor units consisting of 2 to 3 muscle fibers per neuron. 3 cranial nerves provide innervation to the 6 muscles. Cranial nerve 4 innervates the superior oblique, that's the trochlear nerve. Cranial nerve 6 innervates the lateral rectus, that's the abducens. And cranial nerve 3 innervates the remaining 4 muscles, and that's the oculomotor. Strabismus, known as lazy eye, is a disorder present at birth where the eyeballs are not properly aligned with each other, and this can lead to double vision, which we term diplopia. Sometimes we have a difficult time remembering the extrinsic eye muscles and the cranial nerves that talk to them, so you can use a mnemonic device that utilizes a chemical equation analogy to remember these relationships. LR6, SO4, all the rest are 3, right? Lateral rectus, which is innervated by cranial nerve 6, and then superior oblique, innervated by cranial nerve 4, and then all the rest are oculomotor, which is cranial nerve 3. Okay. Looking at the eyeball, it's a hollow sphere that consists of an outer wall that surrounds several chambers and supports the lens, which helps to focus light as it enters the eye. There's 3 distinct tissue layers that maintain the shape of the eyeball and transduce light into electrical signals. They include the fibrous, the vascular, and the neural layer, and you can see each of them here in the cutaway. Okay, fibrous layer consists of the cornea and the sclera. The vascular layer consists of the choroid and the ciliary body and suspensory ligaments, and the neural layer consists of the retina and the optic nerve. The fibrous layer is the outermost, and it's made up of 2 components: the sclera, which is the white part of the eye that covers nearly the entire eye, made of irregularly arranged collagen that allows it to resist deformation from external or internal forces to maintain its shape. It's an example again of the structure-function-core principle that we talked about in A&P 1. The cornea, which is continuous with the sclera anteriorly but is translucent instead of opaque due to parallel arrangements of its collagen fibers. It is avascular, and this allows light to pass through the eyeball without being scattered. Okay, so because the cornea is transparent due to the regular arrangement of the collagen and the keratin fibers in the extracellular matrix of the cornea, when light hits it, it bends towards midline, and this is called refraction. As it passes through the aqueous humor, which is the liquid that exists between the lens and the cornea, it bends even more. When it hits the lens, it'll bend even further. The lens is also transparent. It's made up of a protein called crystalline, which are produced by lens cells that are inside the structure. And then it'll hit the vitreous humor, which is a gel-like substance that bends the light even further. And in a properly developed eye, that light will come into focus in a spot in the retina known as the macula lutea, which happens to be the part of the retina that contains the greatest number of photoreceptors. Particularly the cone photoreceptors, which we're going to find out are responsible for our ability to turn various wavelengths of light into a sensation that is interpreted by the brain as color. The vascular layer is the middle layer, and it lies directly between the cornea and sclera, and it's composed of the following components: the choroid, which is the most extensive component, which contains capillaries and a pigment that minimizes the scattering of incoming light rays; the ciliary body, which is continuous with the choroid anteriorly and has a ring of smooth muscle that surrounds the lens; and the suspensory ligaments that connect the ciliary body to the lens, and this allows for contraction and relaxation and changes the shape of the lens in order to focus light. And so you might wonder, well, why would we do that? Why do we need to change the shape of the lens? Well, if you're trying to focus on an object close to the eye, the light rays bouncing off of that object need to be bent more severely in order to hit the sweet spot of the retina and come into sharp focus, and that requires that the lens become thicker and fatter. And when we're looking at a distant object, the lens can be relatively thin, and we can see the object with relative clarity because we don't need as much refracting power. The iris is the colored region of the anterior eye, and it's an extension of the vascular layer. The amount of pigment known as melanin determines the iris color, and again, this is all inherited. The pupil is the opening found in the center of the iris through which the light enters the eyeball. The papillary sphincter muscle, which is found in the iris, contracts during parasympathetic stimulation. This decreases the size of the pupil and restricts the amount of light that enters the eyeball. The pupillary dilator muscle, also found in the iris, contracts during sympathetic activation and allows the pupil to increase in size and allows for entry of more light into the eyeball. Okay, so remember from last term that we checked these reflexes using the ciliospinal reflex and the pupillary reflex. When you did the pupillary reflex, you were able to shine the light into the eyeball and watch the pupil constrict, and that was a property of the oculomotor nerve. And when you did the ciliospinal reflex, you pitched the side of the neck without giving any warning to the subject, and you saw the pupils dilate, and that was due to sympathetic leads coming to the pupillary dilator muscles, which are postganglionic neurons coming off the cervical paravertebral ganglia. The neural layer is the innermost portion of the eyeball that's known as the retina. It's an incomplete layer found only deep to the choroid and is composed of two layers in the following features. The superficial layer consists of a thin pigmented epithelium which decreases light scattering and nourishes the photoreceptors. Now when we talk about the superficial layer, it's going to be the part of the retina that's closest to the choroid. Okay, so keep that in mind. The deep layer are the photoreceptor cells that form the optic nerve, and the photoreceptor cells are found in the retina. They detect and transduce light into electrical signals. Other features include the ora serratia, which is a serrated anterior margin of the retina between the anterior edge of the retina and the posterior edge of the ciliary body, creates a visible line, and is one of the 2 points of physical connection of the retina to the eyeball, the other being the optic disc. The fovea centralis is a region of the retina that has a large number of photoreceptors and allows for detailed vision and the ability to focus on an object. The macula lutea is a yellowish central region of the fovea and has a large number of photoreceptors, while the optic disc is the location where the optic nerve exits the retina and contains no photoreceptors, and we know this as our blind spot. And in lab, you're going to do a blind spot test where you use a 3x5 card, and you're going to determine your blind spot by holding the card at arm's length, covering one eyeball, holding that card in front of the closed eye, and then looking with the open eye at the image furthest from that eye. While focusing on that image, you bring the card closer to your head, and what will happen is that the other part of the image on the other side of the index card will vanish, and that will be the location of your blind spot. Now, you'll find that the blind spot is in different locations in the right and the left eye, and so the reason we don't have a blind spot in our normal visual field is because one eye compensates for the other when we look out in front of us.